This is Late Night Science with your hosts today, Axel Schumacher and Azam Shagagi. Our guest today is a molecular biologist from the beautiful city of Tübingen in Germany. In his early career, he studied mRNA molecules and finally founded the biotech company CureVac, the most advanced company in the development of mRNA-based drugs such as COVID-19 vaccines, that grew to be a multinational unicorn. Our guest, Dr. Ingmar Hoerr. Ingmar, how are you doing? Welcome to the show. It's so good to have you on board.
Thanks a lot. It's a pleasure. Thanks.
Hey, really nice to see you. Well, where are you isolating yourself?
Well, I'm in my home in Tübingen, Germany, south part of Germany.
Ah, very nice. I love that area. Beautiful.
Yeah. Swabia.
How's the weather there?
Well, just a winter thing, but it's fine. Yeah. So we had some snow, but now it's melting, and it's a glimpse of the beginning of the new season. So it's getting into more light, not that dark anymore. So it's very fine. Thanks.
You are the founder of CureVac, and CureVac is pioneering the next development of the new class of messenger RNA vaccines. If you can elaborate more a little bit.
Well, yeah, that's we're working for more than 20 years on this so-called RNA business. And when we started, nobody was really knowing what does it mean. You know, what is RNA? What is the difference between DNA and RNA? What is the gene? Is it genetic material? Is it not? So RNA is a building block of life. It's a messenger. So it's very important that it's a kind of software we all have in our bodies. Information is encoded on the genes. It's the DNA chromosomes. But how to get this information into proteins? This is messaging. And it's done by RNA. RNA is a very unstable molecule, which is just meant to kind of delivering information to the right place of the cells to make proteins. So it's kind of envelope. So it's quite interesting because once the envelope reaches the guy who is supposed to read it, then the envelope is destroyed, and it's the same for RNA. So therefore, RNA is a perfect molecule, for example, for vaccines. It just needs to get the information to the body, that the body is mounting an immune response. And once there's an immune response, then there's no need anymore for this information. So RNA is destroyed.
I mean, RNA molecules, they are, or were at least in the past known to be very unstable, or at least people thought about this. So how did you solve that problem? And how does it compare, for example, to other mRNA vaccines that are on the market already?
Yeah. So yeah, that's right. So when I started my PhD work at that time, I was very scared of RNA and working with RNA because indeed it's really unstable. So, but I used already naked RNA, so without any protection, and I checked the RNA into the mice, in the muscle of mice, and it worked. So RNA was uptaken by muscle cells, and also the protein encoded on the RNA was expressed. So therefore, it's not that unstable as you think about it. But of course, if you really want to do vaccines, you need to stabilize the RNA, and people take so-called liposomes. Liposomes are molecules which are laying on the RNA, a kind of fat molecules lying on the RNA, protecting the RNA from all the environment. And also it's mediating the uptake into the cells. That's also very important. So everybody uses these liposomes, Moderna, BioNTech, and also us, which is really important to deliver the RNA. But the course of RNA, how you do this, what kind of sequence you're using, whether you have stabilizing elements or not, this is proprietary technology of CureVac. So we're using also elements which are not to be translated in proteins, but just elements that just stabilize the RNA. So that means it's more temperature stable, and also CureVac needs a lower concentration of the drug as the others. That's the reason why the RNA CureVac is something special.
Yeah, that makes sense. Yeah, I heard like the first vaccine that came on the market from BioNTech, it has to be shipped at basically -80 degrees, which is of course very difficult and logistically it's a huge problem. So how does your vaccine compare?
Well, I think this BioNTech was bound to a fast track thing, and that's fine because it's also needed as soon as possible. You can get to a vaccine. It's really it's important. But CureVac was more bound to science, and CureVac was working a lot on stabilizing the RNA, a lot on temperature stability. So they test a lot of constructs, and then they decided for the construct which is the most stable construct on 4 degrees, and the others they discarded all the others construct. So it takes some more time for this development areas, and now we have CureVac has a product which is really stable. And also, as I said already, low concentrations are also important. So the thing is really with the screening of the right RNA setting. I think this was very important and crucial for this scientific development.
How come they didn't use RNA before in the vaccines, and now it's actually kind of like a trend that we see most of these companies are using?
Well, it's a very interesting question. You know, when we started the company and we also spoke to venture capitalists, nobody was really interested in vaccines. So it was regarded as old stuff, you know, everything is there. There's nothing new here. There's no need for vaccines. So therefore, we switched to oncology. As you know, oncology is really important for especially the Western countries where people get very old. So it's a real threat as well. So we decided to work on oncology, but it's not that easy. You know, you work with body cells which are mutated, which are hidden by themselves. Yeah, so you're not working against any virus or bacterial. So therefore, the approach working against a virus, which is a foreign thing, you know, it's much easier and much more straightforward. But from our perspective, when we founded the company, there was no interest on this virus things or on this vaccine thing. So therefore, we were forced going into the direction of cancer. And now it's a good thing now, you know, that there's lag in this COVID stuff, and I think this technology is really made for vaccines like that.
Mm-hmm. That's great, yeah. So, but you mentioned already, like one other use case is the cancer therapy, right? So, what do you think? So, what other areas is not only CureVac, of course, interested in? What other diseases and in general, where do you see the strengths of the technology? Can you apply it to all kinds of diseases, or do you have also limits?
Yeah, in theory, you can do lots of things. Like RNAs is used for everything in the body. You know, it's encoding a lot of proteins. The same you can do, for example, think about insulin, diabetes, for example. So there's no immune response. It's just the protein, which is very important to work on this mechanism. Or think about EPO. If you want to win the Olympics, you know, just take an RNA pill and you will win. So I'm kidding, but you know, there's an open space for lots of things. You can induce the immune response, then you have a vaccine, or you level down the immune response, then you have some protein drug you can use for any disease you think about. You can also use antibodies. You know, you can encode antibodies on the RNA, so you can have a passive immunotherapy, especially for cancer. This could be quite important. So there's a completely open space. And this was also in the beginning the threat, that we were thinking we can do everything. So we tried a lot of things in parallel, and we weren't really focused. So now we have a focus, and I think this is really healthy for the company.
Yeah, having a focus is always very important, especially if you start out with a company. So I'm talking about starting the company. I can imagine that in the first years, and you mentioned already, it was not easily possible to really push this into the market. So was it difficult to get your company financed at the beginning, or How did that work out?
Yeah, that's a brilliant question. Of course, if you do something disruptive, it's always a real issue. And there are no comparables out there. So when we approach the venture capitalists, they say, "Well, I've never heard about this stuff. Why you think this is so good?" So you had really to count on your own data, and there was no comparable outside there. And it's always easy if you do things others are already approaching. And it was also easy for followers of us, once Dietmar Hopp, the SAP founder, was investing, and Bill Gates Foundation was investing into us, then of course it's easy to follow. But the hard thing is really being the pioneer and being the first, doing something completely weird. And also scientifically, people weren't understanding why this molecule should work. We all know this molecule is really unstable. So what the heck you do that it's working? So I was elaborating I was telling a lot about it, but they weren't really believing in it because this is a paradigm. It was already a paradigm for every scientist. Don't work with RNA. This molecule drives you mad, you will lose. So first thing
but
is,
that's my experience. Never trust your colleagues. Yeah, if you have a good idea, you just have to do it and then see where it goes, right?
Yeah.
I love that story. Yeah.
So how can we use mRNA now for, you mentioned that we can use it for any types of disease now. It's kind of like we have open source in technology. It's also kind of like an open source in healthcare. How could it be, like, for example, used, for example, in precision medicine, which is important?
It's a very important question whether you produce something for everybody or you do an individualized approach, person by person. So, and I like this, especially in terms of cancer. Every cancer is somehow personal. So you can't really match. And I think that's really one of the biggest issues we have in cancer therapy, that one fits for all. This approach is not working with cancer. So our vision is really, together with Elon Musk from Tesla, that we develop printers. Printers means RNA machines which can print RNA in a couple of hours. So, and using these printers, established these printers at the pharmacies, for example. So then every pharmacist can print a special drug for every patient. I think that's the real thing.
Like a 3D printing?
Pardon me?
Like a 3D printing?
Yeah, it's exactly like a 3D print. The pharmacist just needs the information. For example, if you have a tumor in your brain, so the pharmacist needs a sequence, what kind of tumor cells you have. So diagnostic is very important. But once he has this diagnostic, he can then put in the code of an antigen. That means an information for an immune response against exactly these cancer cells. So, and then, cancer cells, they tend to mutate, they tend to change their profile. And this guy is following up these cancer cells. So if something happens there, if you can't really control the cancer, you take the new mutated cancer cells and analyze these guys, what they changed, and then it can easily adapt. If you just change the sequence of the printer, and the printer gets you a new drug, which is dedicated to eradicate this exactly cancer cells which were mutated.
Wow.
Great.
Yeah.
You can follow up. This
is the next level.
It's kind of like.
Go ahead.
So then we go to the longevity concept, isn't it? Wow.
So it's really a personalized thing, and it's a paradigm shift. You don't need pharma industry anymore; it's done in the pharmacies.
So there's even more you want to disrupt. So
it's a very disruptive thing.
But I think we talked a long time ago. We talked about this before. What, in theory, would you need to really tailor a therapy to an individual? I think one problem we have to solve for that is also the data sharing aspect. So the private healthcare data is, of course, usually very siloed and not easily accessible. So what we do at Chivome, so my company, is that basically making healthcare data available of individuals. So would that be helpful to you, that you can say, okay, this is the genetic code of this person, and this is obviously different to the next one, and then we can tailor our drug directly to this genome, right? So
it's very important to know, in contrary to COVID, where you just need one information, and it's since it's COV2, and there are no mutation outside, then it's working. If the thing is mutating, then you have to change things. In terms of individualized cancer therapy, you exactly need to know what type of cancer, and if it's a kind of prostate cancer or brain cancer, every brain cancer is different. So you can't do this as a kind of mass production. So you always need to get the data from each patient, and then from this patient you can use the data, and you can then manufacture exactly the tailored vaccine for this patient. So therefore, it's a completely different thing. What is going on now on the COVID thing? It's a mass production for everybody, but here you can just do a kind of individualized production. So in terms of data, you need, of course, the data of the tumor cell. So you need always tumor data of the patient. Yeah, that's the first thing. You have to do this diagnostic step, and once you have this data, then these data are delivered to the machine, and this machine is producing the vaccine. And then you have to do again, after maybe half a year, you have to do again a diagnostic check and see what happens to the tumor. And for sure, this tumor is going to mutate. It's always like tumors are doing that. So tumor wants to escape from the threat. So therefore, you need to get the new sequence of the tumor, and you do a new vaccine, a modified vaccine, to access these mutated tumor cells. And this can be a lifelong process. It can be adjusted as long as possible. And you can't eradicate the whole tumor. It's not working. Somewhere tumor cells will rest, and they will just stay and rest, and they will mutate, and they will come again. That's the thing with tumors, that you can't really get people really healthy. It's always a side.
It doesn't get eradicated.
You can't eradicate it, really. You can only have kind of sleeping cells, which are there, but not anymore harmful. But once there's a trigger somewhere, then the cells will break out, and they will get again into a tumor tissue.
But I wonder, isn't that a nightmare for regulatory bodies? Like, if you cannot run a classical clinical trial where you get 10, 20,000 people through a trial and see if the drug works, because you're actually working only with one patient. So how do you think that will work?
Well, in Germany, there's a law that a doctor who is able to mix his own drug, he can do that. So there's no approval something, and so therefore it's not a big bar for doctors, since it's a premixed drug, but the doctor has to take his responsibility for this, for sure, So he can't say, well, no, this was a mistake, and so no, I think he has to take care of this responsibility. But if he's doing that, he can do what he wants here, especially for cancer patients, which there's nothing else available. So that's quite easy in terms of the law in Germany.
Right, but you want to go with it, obviously do this all over the world, not only in Germany, right?
Yeah, but it's more really that you take doctors more in responsibilities, and also in the treatments. And then I'm quite sure that the regulations will be, or they are all kind of same around the globe. So therefore, you have to teach doctors to do things, and of course they have to start with a low dose, for example. Otherwise they take a lot of risks in autoimmune responses.
How far do you think that we are from this phenomenon, that it's getting out there? The doctors are kind of like in control seat, and the government is just playing the second party, not the first party in charge. How far are we?
Well, it depends on data. So once you have data that you can really
how much data is enough?
I think it's a kind of a trial thing. You have to do a trial with patients in this kind of individualized therapies. So you need equipment. You need these printers, and where can you install that? So the first important thing is to have the printer ready. And once you have it, I I think it's quite easy to go into hospitals and ask doctors to fulfill the individualized treatments using the printers. So it's more a thing that we have to get these machines from a prototype into a small production scale, and then you can run into the clinics, and then you will get all the data. Because for sure they will get the data, and they're allowed to do so. So on the regulatory perspective, it's quite easy. It's more really that it's a conceptual thing that we have to distribute the printers in many clinics and hospitals.
I heard that it was more like a coincidence that you started working with the company from Elon Musk on this project, right?
Yeah, exactly. So there was a German company called Grohmann. This was an automatization company, and we were in contact, and we also had some small projects here in terms of the printer. And then Tesla was buying these guys, and then Tesla wanted to throw away everything what they did. They did also service for BMW, for example. So, and therefore we went to Palo Alto, and we went to the management of Tesla. So the first meeting, I didn't meet Elon Musk. It was a colleague of him. But the colleague was asking us, okay, he will tell Elon, and then Elon will come to us, and then he will talk about it. So we were really happy that he was open to talk about it. Yeah, and we are the only project of Grohmann which is outside the company. Everything was, inside Tesla.
So what is the bioreactor that I believe he's making a bioreactor for you guys specifically to develop vaccine? Is that true?
You mean Tesla?
Yes.
Well, Tesla is working in the printer, so in the automatization. It's a kind of same technology which is important for Tesla cars. Yeah, so it's all the interface, everything inside. So it's a kind of adaptation of Tesla technology. And the printer is completely autistic. I can't say autistic. Completely independent. Yeah. So it's an automatized process, and nobody can interfere with this process. It's done. So it's like a computer. Once a computer is not working, you throw it away. It's the same for this machine type of machines. So it's clear that nobody can interfere with this printer, and everybody uses some software where you can type in, and then you need to fill in all the ingredients, what the machine is needed. But that's it. So it's an automatized process.
So it's a bioreactor, like literally one giant printer.
Yeah, it's a kind of mixing of fluids, for example, incubation and purification process, and everything is inside in this machine. So it's a kind of a lab guy sitting there. He can do also human, but this is automatized everything.
Yeah.
And what is the capacity? What is how many?
So
what's the outcome?
It's for sure it's for small amounts, but we also don't need much of the RNA actually. So you can think easily about 1,000 dosages, for example. So if you think about a milliliter range, then you get maybe thousands of dosages for a small community of people, for example.
Fantastic. No, I love it. If technologies really come together to build something absolutely new, that's great. Great stuff. And you need obviously you need then the help of other companies as well. You cannot develop everything in-house, right? Talking about having partners, I think you partnered now with several pharma companies, right? So to distribute and work on the novel vaccines.
Yeah. So I'm also very happy, and I'm also proud of it, because it's crucial. As you can see, the biotech Pfizer deal, the momentum. It is clear that a small company as CureVac, it's too much for us. It's completely clear. So therefore, the management was doing a long time already, beginning of summer, that they talked to a couple of companies about partnering opportunities. So it took some time, and not everybody wanted to partner with CureVac, but at least Bayer and GSK, which is brilliant because GSK is a vaccine company. It's the best what we can have.
Yeah, it makes perfect sense.
And Bayer is a quality-driven Germany company, not that much in vaccines, but they are really well known in Germany and have high quality aspects. So it's also one of the best we can have. Yeah.
So basically, you are working already or preparing for the next pandemic. Yeah. So like, I thought that's what you're doing with GSK, right? So basically seeing if there are new mutations or new viruses showing up somewhere in the world, and you can relatively quickly move in and offer already a vaccine quite fast, right?
Yeah. It is really crucial. So, and it's a real threat of the COVID scenario, because once there are some vaccines, what happens if the virus mutates, and the vaccines are not really protecting anymore. So you need really a backbone where you can have, as soon there's a mutation in the virus, you need to have something, how you follow this guy, and you can use this technology, what is in our hand. It's a phenomenon, especially taking 1 year from the first detection of COVID virus. It took for the guys of BioNTech, Moderna, 1 year, and they got their vaccines.
Yeah, that's
amazing.
That's amazing. Yeah, absolutely. To see what we can do nowadays. So I think even the biggest hurdle is not so much even the technological, how quickly you can develop a solution. It's rather than really going through the trials and doing your due diligence basically on what you developed, right?
Yeah. And a major hurdle is the production, for sure. You need really gigantic production facilities. That's also really important. And I'm happy that CureVac now is following up with Bayer and also other partners in doing this.
Yeah, I think. Go ahead.
How long do you think that would take to develop the vaccine with the partners, CureVac vaccine?
Well, it's said it's a second quarter of this year that they're gonna have the first vaccines out there, and they got approval from the authorities. But for sure, it takes more time. So it takes at least 1 year to have billions of dosages available, and then also regarding the mutations, to have a new type of vaccine available. But it takes some time, but once you are there, it's not an issue. You can easily scale up the RNA manufacturing. You can do tons of RNA vaccines if they're needed. It's not a technical issue. It's more kind of timing issue and money issue for sure as well.
So in general, like you have so much experience now building a company from scratch. So I think there's a lot of young researchers out there that would be very interested in getting your guidance in what to do, basically. Let's say, imagine you would have to start again. You're a young student. Do you think you would do something differently now compared to your path, or would you do exactly the same stuff again?
Well, it's a very good question. It's always interesting to look from behind back into the early days, and you're not experienced, if you don't have any experience. So it's very hard to think about some guys who are just entering into the business coming from the university. Well, I think what was a kind of mistake that we were trusting too much on people, kind of business angel guys, they were really cheating us. Yeah, so it's easy for these guys because we don't know. We were happy to talk to these guys, and they told us, we give you hundreds of thousands of euros, for example. You can get things started, and you don't have to take care of. But we try really to get VC capitalists. We help you in the VC, in the business planning, in business plan writing. We do this, and the thing was that we weren't really carefully listen to the guy. So the guy was giving us money, and there was no contract, And I was thinking, well, it's on this guy. The guy should look how he can secure anything on the contract, because I wasn't aware that it was my mistake. I can't just get money from somebody without any contract. It's my fault.
And then he would take the ownership?
Yeah, I don't know. He didn't want to take the ownership. We were saying, well, it's about 20% or something like that. So it was also kind of communication. And then I was giving my bank account number, and then the next day the money, the 100,000 was there in my bank account, And
Wow.
I wasn't really thinking about, I was thinking, how nice of this guy, the guy is trusting us, delivering his money on our bank account. But in a legal perspective, it was wrong by me, but because I can't really receive money from somebody without any obligations, it's not possible.
Yeah, but to be fair, like if you are coming from the academic environment, usually most scientists have absolutely no idea how to really start a company.
Yeah.
So retrospect, would you say it would make sense that as a young scientist and you have a great idea and you want to start your company, maybe wait a few months, learn first basic business skills, or how would you go about it?
Well, I think it should be kind of dedicated VC company, not a kind of business devil guy. So be careful. And if you have a guy, you should really know it and write, and you should take also some law assistance, because you should know about it. What's the background? Are you obliged to pay the guy the money back because it's wrong somehow? This is exactly what the guy was doing with us. He was trying to get some more money. We were doing a roadshow, but it was not running very well. So the guy, he gets scared, and then he wrote me a letter from a lawyer office. I want to back my money. Yeah. In 2 days, I want to receive my whole money. Otherwise, I will prosecute you.
Wow.
So and we already were spending that kind of money for equipment and things like that. So we weren't able to do that. So the only rescue was the local bank in Tübingen. So we went to the local bank, say, well, we're squeezed. We have really an issue. We need 50,000 euros from you guys. Yeah, you should help us, and we will pay back. We are all academics, so we will get a job somewhere, and we can spend it. Yeah. So they were believing us and say, well, we help you out here. Yeah, and we really believe in your names. We will somehow be able to do this 50,000 euros once in a while.
It's nevertheless, it's a great story. I mean, and of perseverance. So from the mindset, do you think it's really important to learn these things, to go through these valleys in your entrepreneur career, or what did you learn from it? I mean, or what can you teach the young entrepreneurs, and how can they shape their mindset to be successful?
Yeah, it's I think it's an individual thing. Everybody has to go his own way, and everybody's doing mistakes, as you say. Well, mistakes are also somehow helpful, because you then avoid the second time the same mistake. Yeah. So I think it's an evolution process. Everybody has his own evolution. The thing is really, don't trust too much people you don't know. You should only trust people you know, or some people who have some reputation somewhere outside the thing. So if there's nobody with reputation, don't trust this guy. We should understand what's the mission of this guy, why this guy wants putting his money into your business, what's the real thing behind it. So don't be naive. I think that's the thing. Try always to be on the safe side. Try always really to understand the impact, what's going on there, and try also to be professional. See really, whether you can trust your business plan. Our business plan was crap. We were writing stuff we didn't know. We were writing about clinical trials, never conducted any clinical trials. And so the prognosis was completely wrong.
I think the dynamic has changed right now, because now we can see the teams are more diverse with the business background, scientific background, and technology background. And I think that has changed. But how do you feel like now that you have, invented this amazing technology and science that is going to actually help the world, and billions of people around the world. What is the feeling like?
It's
Express.
well, my both legs are on the ground, so I'm not really jumping. First of all, I will really see that CureVac is getting into the approval process. That's very important for me. And then, of course, it's really a great thing thinking about it. But the thing is really, I was always a believer. I was always knowing about this real impact. I didn't know about COVID, clear. But from the very beginning, I was completely energetic about this technology. And this was not always good, in some of these investor calls, I was exaggerating a lot of things, and they would say, well, this guy completely mad. But in the end, I'm really proud of things, what's happening. I'm also proud of all the collaborators, about my co-workers, about the company spirit, that we never gave up in these really dark times. So I'm also proud of Dietmar Hopp, that Dietmar Hopp was following the track, without lots of data. He was just believing me. So also to have this personal relation to people like Dietmar Hopp or Bill Gates or Elon Musk, this is something I really think this is great. Yeah, and this is an achievement for sure. It's even more an achievement than what's going on in terms of the data right now. But this is now business as farmers are used to do so. But building companies, it's really, really, really difficult.
I know your name goes down the history for sure, along with all these like great pioneers.
Yeah, hopefully. But I'm not dependent on this,
Of course, an inventor is always looking for the society and the social impacts. Obviously, not himself, right?
Yeah.
And you mentioned that this year you were experiencing some health issues. Would you be comfortable sharing those?
Well, it was an aneurysm in my brain, so there was bleeding in my brain, and I was unconscious, laying in the hotel room. I was in Berlin at that time, talking to the government. This was the debate, of Trump acquiring dosages from CureVac and things like that. So it was a kind of emergency thing. And I was just only 1 or 2 years, only 1 or 2 days, I was becoming a CEO again. So I was the head of supervisory board for 2 years. And then I was jumping back on the gears of CureVac. And this only 2 days. And then there was the kind of knockout. And so I was then fighting for my survival. I think this was the biggest challenge I got in my life. Yeah.
Was there a reason?
It's a kind of my father is healthy. It's not in my family, so I don't know. It could happen. Yeah. It could happen, and of course it was stress. It's clear that my blood pressure was high at that time, so there were many things coming together. Yeah.
But you went through it pretty good, so this is amazing. It's maybe
we're happy to have you.
The mindset, right? No?
Yeah, and also support. You need a lot of people supporting you, like always. So I got really support from a lot of friends, from my wife, for example, from my family, from my parents, from everybody. And this keeps you going. Yeah. And the other thing is I never give up. It's also in my mind, in my brain.
Exactly. Never give up.
Fighter. Yeah.
Never. So for everyone who's listening, never give up.
Never.
Not repeat that enough.
Yeah.
So from scientist to scientist, or before we close here, but where do you see science going in the next years? I think we have so many changes going on right now, like so many technologies coming within a very short period of time, changing really how we do medicine and all the other stuff, also in self-driving cars, quantum computing, and nanotechnology, so many things coming. Where do you see the biggest changes in the coming years?
Well, it's as you said, it's a kind of information driven business. It comes more to information and less on drugs itself. So therefore, I see a really a strong move in the direction of the RNA technology, where you give information to the body, and the body is then producing its own medicine. And I think that's a real disruption. What's going on? And COVID is just a start. It goes in all directions. In my eyes, it's kind of Alzheimer, tumor thing, many things, and it gets normal somehow. Yeah, maybe even for children that they take some RNA for something. I don't know. Yeah. So this is this information-driven treatments. This is the disruption, the real disruption in the settings.
That what are the next?
Yeah.
Sorry.
No, please.
So, what are the next big trends in actually in healthcare that you would say? Some say that it's genetic engineering, mRNA, CRISPR, but which one do you think that would actually be in the direction of humankind? And obviously, we're looking into exoplanets, like space exploration.
Yeah. I think it's really important to gather as much as possible information about health issues, so that you really see how you can interfere, what you can do, and then, as you said, there are a lot of technologies out there, CRISPR, siRNA, messenger RNA, how you can interfere, how you can build drugs, and I think this is a real information-driven business. It gets more and more into the information, and not only in this just on the drug making process. So drug making itself, the pharma business industry as it was the last 50 years, I think this will fade out in more in this typical printing, personal treatment thing. I'm pretty sure about it, yeah, that everybody will have his own printer, maybe at home installed, you know, where you get a recipe from the doctor and you can just do it by yourself, more or less, you know. Could be like this.
Yeah, that's amazing. Great. Maybe have a great future, healthy future in front of us now.
Hopefully, yeah. Hopefully.
Any other comments or messages you would like to?
No, what I said, it's far beyond COVID. It's far beyond. It's just the start of everything. And this is real interesting. I'm thrilled at that this is a paradigm shift. So I was always telling over the beginning of CureVac, we shift a paradigm, and people were laughing. They were, what kind of paradigm? What do you think? You know, why you think like that? You know, what did you achieve in your life? Why you think you can change a paradigm? You know, and now
then
this
COVID
is really
wasn't
happening.
a bad thing for you guys. So then COVID wasn't a bad thing for you guys.
Well,
it was.
Of course, it's a bad thing for everybody.
I know,
you know, this is nobody.
Different.
Nobody wished the kind of things like that. But I'm happy that we can. All the RNA thing, or all the RNA guys, they can provide a solution for this. And this is an ignition for completely for the paradigm shift. You know. So for this kind of view, I think it's positive. Yeah, it will create a paradigm shift. I'm no doubt about it. I've no doubt about it anymore. Yeah. So this is the good thing, but the bad thing, of course, is the COVID scenario, and it's a real crisis. And I don't know whether we have to live with this, you know, for all life, for the whole life, or whether we can irradiate it like the pest, like the other big issues in the past of the history.
Yeah. Thank you so much. This is super exciting. So I will follow CureVac for sure then over the next years and the mRNA technology. And I have to read much more and then see how we can help all these businesses with the data information gathering in this field. So thank you so much. This is really very interesting.
Thank you so
Assam,
much.
do we meet in Paris anytime again?
Of course, yes. Well, that's what I was saying, that hopefully you recover very fast, and the next time we meet somewhere in Europe.
Mm-hmm. Exactly.
Yeah, definitely. Yeah. Wish you all the best and the recovery. Send you all the positive energy, and stay strong. We all support you.
Thank you very much. Thank you. Pleasure.
Thank you.
Thank you so much, everyone.
Bye-bye.
Bye.
Okay, podcast is again done. Wonderful. Hey, you are still here. No. You know, the podcast is over. You could go home, or if you are home, you could go outside. And you know what you could do if you're outside? You could get vaccinated. Yeah, because vaccination works. I'm sure you want to know how a vaccine works, right? Yeah, you do. Look, since you're already here, I can tell you how an mRNA vaccine works. It's like this. Imagine every virus is different. Like us, we all carry a different genetic code. And the virus that causes COVID-19 also has its own very unique genetic code. We scientists, we take this code apart and we look at the mRNA, that's the messenger RNA. We just talked about it in the podcast about this molecule. This mRNA tells our cells what to build, that are proteins or enzymes. So what we do, we have to get this mRNA information, the code, into the cell. Yeah? Imagine this is the cell. So we coat the mRNA with lipids so that they can easier enter the cell. They go into the cells by injection. That's the vaccination process. The mRNA in the cell then tells the cell to make a specific part of the virus, which is called the spike protein. The spike protein helps the virus usually to enter the cell in the first place. Yeah. So what happens then is our immune system can produce antibodies and T cells to destroy the spike protein. As a consequence, the virus cannot reproduce anymore. It cannot infect new cells. In the end, we are protected. So if you're exposed to a new virus in the future, your immune system knows already what to do. The new viruses are quickly recognized, the spike protein is destroyed, and the antibodies and the T cells are ready to really destroy all of the bad viruses in your body. Without a vaccine, your body has to identify the virus first, learn how to fight it, and carry out the immune response. In the meantime, the virus can replicate to a level beyond what your immune system can handle. So, which means you will feel sick. With the vaccine, your body can more quickly identify the virus and skip straight to starting its immune response. So get out there and get a vaccination. Yeah, but before you go, please remember, if you like the show, please leave a like, subscribe to the show, and if you watch it on YouTube, press the bell icon so that you get all the notifications of future podcasts. Thank you so much. Have a great time and stay safe.